Hi, this is Miss Litton, and this is still my wonderful honors bio, period 4. Say hi. Hi. Now slightly full with pizza. And we're going to continue on in chapter 25, control of gene expression. And we have discussed prokaryotic control, so now we're going to move on to eukaryotic control. So the first thing we need to go over is keeping in mind that we're opening sodas. Keep in mind, in some cells, some genes are always turned on. These are called housekeeping genes. They're needed all the time. So there's nothing that's inhibiting their production. But things like hemoglobin, both red blood cells, muscle cells, and pancreatic cells have the gene for hemoglobin, but only red blood cells turn on that gene. Just like only insulin genes are only turned on in pancreatic cells, and myosin is only turned on in muscle cells. So different genes are turned on. And the other thing we want to remember is all the points of control. So starting at the top with the light-shirted bio buddy, alternate through. We've already introduced all of these points of control. Go ahead.
I'm glad you had to do the whole thing. So it's like translating. It's like the first step of translational control. So it's the process of mRNA.
And this is a good picture, you guys. This might be one that you would consider keeping in your notes, right? There are 5 points of control we're pointing out. How many of these points of control occur inside the nucleus?
3 of them.
3, and 2 outside of the nucleus, yes? And remember, we have what's called differential gene expression. In different cells, different genes are expressed, and in different times. So go to your introduction. Most cells have a complete complement of DNA, all the same genes, all the same genes. Different genes are turned on in different cells, called differential gene expression. Turned on in different cells, called differential gene expression. Slate, take care of number 3. We've actually already discussed it.
Oh, they're just not typing. My partner's not deciding to type.
Okay, now, let me just give you a little bit of an opening on this one. Before, in prokaryotic cells, most of the control is considered negative control. It's about a repressor. Is the repressor on the operator or not? That's called negative control. In eukaryotic cells, it's more about the positive control. There are no operons, okay? But there are things called transcription factors. And transcription factors help RNA polymerase to bind to the what? Promoter. And if those helpers aren't there, then RNA polymerase can't bind. And another factor is there's so much more DNA in a eukaryotic cell. A lot of times the DNA is tightly coiled and put away. It's not even accessible. It doesn't matter if there's RNA polymerase there, if there's a transcription factor there. It's put away. That DNA is tightly coiled on histone protein, so it's not even available for RNA polymerase. That is something called epigenetic inheritance. Epi means above. And so you could have, let's say for instance, for pea plants, you could have a gene for tall and you could have a gene for what? Short. So you have both of those genes in your DNA. But if the tall gene is hidden away, if it's so tightly coiled and put away that RNA polymerase can't even bind to it, then you have an issue because you inherited that DNA from the parent, but you're not expressing it because the DNA is not available for expression. Do you see the difference? It's not like you're missing the gene, you just don't have access to that gene. There are some women who have expressed themselves as women their entire life, and they come to find out that they had a Y chromosome all along. And a Y chromosome would make them what? Male. Regardless of your gender identity, where you see yourself, okay? Chromosomally speaking, they were a male. They had a Y chromosome. But because the Y chromosome was never expressed, then they were a what? Female. Yes?
Is that why sometimes you can have like blue eyes if one prefers brown eyes?
Okay, we haven't done all of the, we're gonna learn all about that. Then I'm gonna explain inheritance like that once I have more time, because I gotta keep going on this. Yes?
Does that happen often, or is that like a rare?
No, that would be a rare thing, yeah. But epigenetic things do happen all the time. That is normal. The male-female part would not be. Okay, so we are going to start with the first level of control, and that is the DNA even available? The DNA is wound on what's called these histone proteins. And if it's all wound up tight, it's not available. If it's unwound, it is available. And that is a normal point of control. When you look at a nucleus, and here's the nuclear envelope right here, you have dark staining and light staining DNA. The dark staining DNA, the heterochromatin, is tightly put away. It is inactive DNA. The one that doesn't stain, the lighter colors, that is active DNA. Now, it can vary due to your age. So for me, I'm not growing any taller. So genes for growth are what? Heterochromatin or euchromatin for me? Heterochromatin, tightly coiled and put away. But for you, the genes for growth could be euchromatin and could be getting expressed. What is this dark thing inside this nucleus? Nucleolus. Okay, that's a nucleolus. So you can see an example of this is going back to females. Females have 2 X chromosomes. Now, within each of our cells, one of the X's condenses. One of the X's condenses and is known as a bar body. It's like a little dot inside the nucleus. And the other X is expressed. Well, in females, you don't know in each cell which X is going to condense. It could be a different one in every single one of your cells. Now, males don't have a bar body. The reason why they don't have a bar body is they only have one X, so they don't have a second one to condense. But females do. So you can see here, they just randomly colored the 2 X's, one red, one black. So you could have a black X or you could have a red X. And you can see those bar bodies within the nucleus. It appears like a little dot inside the nucleus. Okay, that could be one way, if you got a set of cells, to know whether that came from a male or a female, whether or not it had a bar body in it. Okay, because if it has a bar body in it, it must be a female or a man with Klinefelters, but we'll learn that later. Okay, now, where you can see this evidenced is in a calico cat. Only calico cat coloring, where it's modeled like that, is only in the females, not in the males. Because the gene for the coloring is on the X chromosome, so depending on which X condenses, depends on what color you would see. So only the female cats have that calico color. Then sometimes I have people ask me, well then where did more calico cats come from if there's only females? Well, in the males, they're either tan or they are black because they only have what? One X. So you see the mottled color just in the females. Okay, I'm just making sure you understand that. All right, so go to your notes where it says chromatin or chromatin there, DNA and associated proteins. Remember, DNA is wound onto histone proteins. The more tightly wound, the less accessible for transcription. The more tightly wound, the less accessible for transcription. On your examples, slate, you do 2A, and B, blue, you do 2B. Go.
Heterochromatin is a dark staining inactive, so it's tightly wound. And there's also a light
All right. Now, the next one we discussed about transcriptional control, our next level in. We already talked talked about this on the simplest of forms in a prokaryotic cell. An RNA polymerase binds to the promoter and you have operons. Okay, you just don't see this in eukaryotic cells. There's a little bit more to it. Take a look at the picture. Start at the top. You still have a promoter and you still have what? RNA polymerase. But now you have this other region. Can you read that? It's kind of tiny. Enhancer. So you have to have this transcription activator bind to an enhancer, then a transcription factor. And can you see that there's 3 different colors in that transcription factor?
Yeah.
So this is a conglomerate of several other items that all have to be bound together that then bind to the activator that's on the enhancer. And then this grouping all together helps RNA polymerase bind to the promoter. So we're not about negative control and whether there's a repressor or not. You just need A, B, C, D, E, and F things before RNA polymerase will bind to the promoter. Okay, and this is a form of what's called positive control. It's not about the no, it's about the yes. And where these items come from, this is connected to cell signaling. So let's say you have the signal bind molecule, it could be a hormone, binding to a what? Receptor, which is then starting a whole signal transduction pathway, which is triggering this transcription activator, which is then binding on the DNA, et cetera, in order to make your protein. Now, this is a very simple cell signaling pathway, and this is more the reality. So when I say you are beautifully and wonderfully made, and the complexity of your body is, okay, these are the calls that are going through, okay? Several in order to control what genes are getting expressed. So on transcriptional control, most critical level of control. Most critical level of control. Transcription factors are proteins that assist RNA polymerase to bind to the promoter. Blue, you do 2.
Transcription activators, proteins that bind to the enhancer region and usually further upstream. They must bind to the transcription factor.
So start to do 1 cup.
Transition factor to help RNA polymerase.
Oh yeah, transcription activator protein. Usually for
All right. Post-transcriptional control. You already know what this is. Put on a cap and a tail and cut out the introns before you leave the nucleus. So you can have different combinations of exons and you will get different proteins. So post-transcriptional control, differential excision, removal or inclusion of introns, and splicing. And then speed of transport of mRNA through nuclear pores. Through nuclear pores. Slate, use your notes and discuss translational control. Go ahead, slated one. During the stage and blue, post-translational. Go ahead.
Following translation, only a function.
Okay, now, because we had pizza, I'm not going to have you take turns on this slide. I want to discuss this part with you to make sure you have it down. So the first level of control, we talked about chromatin or chromatin. Okay, and this is all about the DNA. Is it available or not? What was the example we saw where it wasn't available and different colors got expressed? What was that? The bar bodies.
Yes.
Okay. Then you have transcriptional control. This is about making what? Guys, I'm stitching it all together for you right now, so get here. Okay, transcriptional control. What kind of things are associated with transcriptional control? mRNA. Getting what? Getting formed. Who forms it? How do you form RNA? RNA polymerase binds to the promoter. Maybe a transcription factor has to help you bind to that promoter so you can make your mRNA. That's transcriptional control. Following transcription, you call it post. What kind of post-transcriptional things do we do? Cap, tail, cut out introns, leave the nucleus. Good. Translational control is all about hooking up with a ribosome and making a protein. Post-translational control is about folding or activating. Okay? So know that when you answer these questions. And as you're answering, you can pack up. Just stay in your seats, but you can pack up. The first 6 people who get this right can put their Chromebooks away once I stop. Wait, I want to hear you. I just got to finish this. All right, Bobby, Jackson, Jacob, Cameron, Alyssa, and Saket. You can put yours away. Okay, stop. Whoa, wait. Cameron. Oh, yeah, Cameron's another. Okay, name the type of control. Most of you got that. Post-transcriptional, because you're talking about cutting out the introns before you leave the nucleus. Okay, next question.
I can't answer first.
Cameron, Mason, Natalie.
Mine's off.
Why is it off? Okay, maybe that's a reason why we don't, do that. Okay, so Sarah, Ryan, Ben, Jacob, you can put your Chromebooks away. And Jennifer. So 3-dimensional folding is post-translational.
You better be after 6. You're always in the top 5.
Bar bodies is all about chromatin. Bobby, David, Wayne, Skylar, Katie, Jackson, Jacob. You can put yours away if you haven't already. What's the ability to bind to a ribosome?
That's
Translational. And then we'll pick up cancer next time. Good job, guys. Hey, guys, put a pizza box in the trash for me. Fold it so I'm not. Make good choices. You're welcome, sweetheart.